Hello everybody. In this lecture, I'll talk about the somatosensation. As you already know that the somatosensory system is different than the special senses. In case of somatosensation, you have the receptors throughout the body, widely distributed receptors in the body, as well as the receptors are activated by different types of stimuli. In case of the special senses, the receptors are located inside the specific organs, right? For example, the photoreceptors are in the retina of the eyes. The hair cells are in the cochlea of the ear. The nasal epithelium inside the nose for the smell receptors, and the tongue, taste buds in the tongue are for the taste or gustation. The stimuli for the somatosensations are different, and there are different types of somatosensations. We'll talk about that. Somatosensation includes touch, which is the tactile sensation, pain, nociception, and temperature, thermoception, and the sensations in the joints and muscle spindles. Those are called proprioceptions. So those are different types of somatosensations: nociceptions, pain, tactile, touch, thermoception, temperature, and proprioception, sensations in the joints and muscles, movement. So those are the somatosensations. Now, for different types of somatosensations, we have different types of receptors, and they are activated differently, and the stimuli are also different, right? The pain, touch, sensation of temperature, and proprioceptions give a very good information about the environment and help us to protect ourselves from the external as well as the internal harms. You know, the pain sensation, if something bites from outside, that's the external harm. But if any lesion occurs, infection or injury occurs inside the body, also you feel pain. So that is also a good, protection, that you know that something is happening, right? So both external and internal environment. Also, touch plays a very important role in our emotion. You already know that how a touch can comfort a person, right? So that is a big part of the somatosensation and its significance for your survival. Anyway, so the main organ that covers the whole body is the skin. Okay, so skin is the main organ in which you have the receptors. Somatosensory receptors are mostly located in the skin. Skin is a huge organ. It is pretty thick covering of the body, and the size is very big, right? We have about 10 to 20 square feet of skin in our bodies. And if you see the structure of the skin has two layers. outer layer is the epidermis and inner layer is the dermis. Epidermis is outer and it is thin, and dermis is the inner, which is much thicker. Okay, and under the dermis you have another layer that is called the hypodermis, which is not a layer of the skin. Okay, we don't consider hypodermis as a layer of the skin. It contains a lot of fat or adipose tissue. Hypo means under or below, right? Hypodermis, under the dermis, and it is not a part of the or layer of the skin. So skin has two layers: outer epidermis, inner dermis. And in the upper portion of the dermis you have the edges like this, finger-like structures. Those are called dermal papilla. Okay, and dermis itself has two layers formed by two types of tissues. The outer 20% thickness is given by the capillary layer of the dermis, which is formed by loose areolar type connective tissue. So soft connective tissue, loose type connective tissue. And inner 80% thickness of the dermis is given by the reticular layer. Okay, reticular layer, which is formed by the dense irregular type connective tissue. And the receptors for somatosensations are located both in the epidermis and the dermis. Most of them are located in the dermis, some of them in the outer part of the dermis, and some of them are in the inner part of the dermis. Based on the type of sensations, the receptors, we divide them into those four different types as I mentioned. One is called nociceptors, are the receptors that receive the pain. Tactile receptors for touch. Thermoreceptors for thermoception, for temperature. It could be cold or hot. So thermoreceptors, there are two types. Okay, cold receptors and hot or warm receptors. And then you have proprioceptors. So those are the receptors. Based on the type of signal, activate them. Okay. Now there is another way to classify the receptors, somatosensory receptors, based on their structures. So these are based on the functions, and based on the structures we classify the somatosensory receptors into include the Meissner corpuscles or corpuscles, then Pacinian, also called lamellar corpuscles, and these two belong to first adapting receptors. Okay, so based on the type of response, how they respond to the stimulus, we divide the receptors into fast adapting and slow adapting. Slow adapting includes Ruffini, and there is another type, those are called hair follicle receptors or hair root plexus attached to the hair follicle. Okay, and there is another type of receptors. Those are called Merkel's receptors. Okay, this one and this one, they are adapting slow adapting and this one, the hair follicle, it could be in between, so it usually we don't consider that hair follicle receptors neither as fast adapting or slow adapting, although some places you will see they consider that as a slow adapting. It doesn't belong to any of those, fast or slow. So two fast adapting Meissner corpuscles and two slow adapting Rufinus cells and Merkel's discs or Merkel's cells or receptors, slow adapting. Okay. Now slow adapting is also divided into slow adapting type 1 and type 2 and fast adapting, also fast adapting type 1 and type 2. We'll talk about that soon. Okay. Okay. Now somatosensory receptors can be characterized by three attributes. One is type of stimulation that activate the somatosensory receptors. For example, pressure could be soft or crude, vibration, temperature. Just few examples I have already mentioned. All of these. That is one thing. Another important characteristics of the receptors: size of the receptive field. So the size of the receptive field varies in different receptors. The size is different, as I mentioned. There are different types of receptors, and if you measure their receptive field size, you see that Pacinian corpuscles receptive field size is smaller than another type, and another type is bigger than the Pacinian. So even within the same type, we see the size could be different based on the location of the receptor. For example, the touch receptors are the receptive field size is much smaller in the fingertip than the other parts of the body. Make sense? So even same type of receptors, their receptive field size could be different. And slow adapting type 1, slow adapting type 2, also fast adapting type 1 and fast adapting type 2. One important information about slow adapting and fast adapting is that fast adapting receptors are located inside the capsule, that means encapsulated. So if you see, this is the nerve ending, okay, and your receptors are here, located here, but the receptors are encapsulated inside the connective tissue membranes. Sometimes the membranes form many layers. So this is called the capsule or corpuscle. Okay. For example, Pacinian corpuscle, if you see, the receptors efferent nerve and the receptors are located inside the layers of connective tissue. Okay, that is for the fast adapting receptors. But in case of slow adapting, you will see the receptors are located uncapsulated or uncovered. So you'll see this is afferent fiber or afferent nerve fiber, and these are the receptors. Okay. So those are three important attributes about the receptors. Okay, multiple dimensions in the somatosensation. When you use your somatosensation, several channels get activated simultaneously, and that is called haptic perception. That means, just think that a good example is if I ask you to touch without seeing. This is an ice cube. Okay, if I ask you to hold it with your fingers, you put your fingers around it, right? And you not only feel the temperature, you of course feel its temperature, which is cold. That is one very important clue attribute. Another is so that will go to your brain through one channel. That is one type of information. Also, you will feel the shape, size, edges, texture, right? The surface, if it is hard or soft. You see a number of somatosensory information you perceive at the same time, simultaneously. Its shape, is it square or round, right? Edges, you feel if it is hard or soft, right? The texture, if the surface is smooth or rough, and temperature, all those information are going to the brain through different channels. Okay, and that is one important information about somatosensory perception, and that is called the haptic perception. Okay, now in this picture you can see this is the epidermis here that I showed before, outer layer, and this is the dermis, which is much thicker than the epidermis. Now you see that most of the receptors are located in the dermis. Some are located in the outer part, that means close to the epidermis, and some are in the deeper part of the dermis. Okay, now let me show you those receptors. I have already mentioned their names. So you see here, these are the papilla, right? Dermal papilla, and here you can see the Meissner corpuscles. So Meissner corpuscles are encapsulated, right? So they are fast acting that I mentioned before, and type 1 fast acting, located superficially close to the epidermis. Okay, and then let's see fast acting type 2, which is Pacinian corpuscle. Here you see fast acting type 2 Pacinian corpuscle, heavily encapsulated. Here this is the afferent nerve fiber, and you have the receptors attached to it, right? And this white structure is like many layers of connective tissue. Okay, so that is the Pacinian, located in the deeper part, fast acting 2. Okay, so those are two fast acting and their corpuscles. Now you see another type of receptors, slow acting, right? SA. So let's see the slow acting Merkel's cells I mentioned before, slow acting type 1. And Merkel cells are located, you see, just here in the dermal papilla. Okay, here. So Merkel's cells, superficial in the dermis. and slow acting type 2. Slow acting type 2 includes uh, ruffini. Okay, that is one type of slow acting, SA2 type 2 here, not capsulated. And another type I also mentioned here, receptors. This part of the hair is called the hair follicle. This is the shaft, and this part is the follicle, and this is the root. So you see the nerve fibers encircle the hair follicle. Okay, these nerve fibers are also known as hair root plexus. Same thing, hair follicle receptors. Okay, and they are activated by the movement of the hair, by the deflection of the hair. So those are some common types, and also here you see free nerve endings. Free nerve endings. Okay, pain and temperature receptors are located there. So those are different types of receptors. Again, we classify them based on their structure as well as based on their response type, and also we classify them based on their functions. For example, thermoreceptors, nociceptors, right? Based on the function, what kind of signal stimulate them, and based on the response includes fast adapting, slow adapting. In case of fast adapting, you will see when you present the stimulus, the receptors will produce burst of action potentials. Then you are keep presenting stimulus. This is the stimulus. The entire time the stimulus is there. However, the receptor will produce response at the onset of the stimulus, and then when you withdraw the stimulus, receptors will fire. However, during the entire period, no response. We talked about this type of neurons. So this is fast adapting. And slow adapting, you will see the neurons continuously, producing action potentials. Although at the beginning it could be more, then it gets slower. However, the action potentials or firings are being produced continuously as long as the stimulus is there. Okay, so that classification is based on the response. Here again, the same thing that I just mentioned, based on the firing, fast and slow. Fast adapting 1, Meissner, 2, Pacinian, slow adapting 1, Merkels, and slow adapting 2, Ruffini. Okay, so these are the main mechanoreceptors. Now we'll see a few of those receptors, how they look like and how they are activated. Okay, so first let's see the first adapting 1, type 1, Meissner corpuscle. Okay, you can see here, this is the actual photograph here, this one, and here it is a drawing, so you can see the structures shown here. Okay, so this is the connective tissue covering capsule, and inside the corpuscle you will see the flattened cells. Those are also called laminar cells. Okay, so you can see these blue are the cells and present like flattened sacs like this. and they are located within the dermal papilla that I showed you in last picture near the surface of the skin, okay, or outer part of the dermis. And their long axis is perpendicular to the surface of the skin like this. You see, the long axis is perpendicular to the surface of the skin, and these are the laminar cells, flattened cells, and they are stuck together, piled together like this, okay. And the yellow structures inside, in between, you see, these are the nerve endings, spiral terminals of afferent axons or nerve fibers, okay. And how these nerve fibers are activated, these afferent nerve endings are activated by a force applied to the dermal papilla, okay. So if you press, that force will do what? Will move, displace the laminar cells sideway. So like one will be pushed this way, another will be pushed that way, like this, okay. So that kind of displacement would activate the adjacent afferent nerve fibers. Make sense? So that shearing force distort the membranes of the axon terminals, okay, located between the laminar cells, and that would open the sodium channels. So sodium will get in, and that will cause the depolarization at the axon terminal. Make sense? Here you can see those laminar cells if you look carefully, okay. Mechanoreceptors, first acting type 2, Pacinian corpuscles. In Pacinian corpuscle, you will see the afferent nerve endings that contains the receptors are encapsulated by many layers like onion, okay, many layers of laminar cells. And when the force is applied to the tissue overlying the Pacinian corpuscle, since these are located in the deeper part, they are activated by vibration. So sustained force on the Pacinian corpuscle is transformed into a transient force on its axon terminal. Slow adapting type 1, Merkel's cells. Distortion of Merkel cells result in the release of a stream of neuropeptides. So the Merkel cells are activated by the stimulus, and that stimulus will release the neurochemicals or neuropeptides at the synaptic junction. So now let's see here. In the Merkel's, you will see the vesicles, and the vesicles release the neurochemicals here in the synapse, okay, and that will open the sodium and potassium channels to cause the depolarization. Rufinase corpuscles or cells are activated by the temperature, change of temperature, and also they are activated by the stretch or pressure, okay. So there are many collagen fibers around the afferent nerve. If you present, the stimulus that is stretching type stimulus, that would stretch the collagen fibers, and as long as the collagen fibers remain stretched, the axon terminals, remain compressed because the axon terminals are intimately attached to the collagen fibers. Okay, and these are located in the deep within the dermis. Now, free nerve endings. I also mentioned that we have free nerve endings in the dermis, and those free nerve endings are mainly for the pain. Okay, and also some of them are for the temperature. So now the pain signal or nociception would stimulate the free nerve endings, and the fibers, afferent fibers, could be two different types, and nociceptor can be divided into two types based on the nerve fibers, afferent nerve fibers, and one nerve fibers are myelinated and another nerve fibers are unmyelinated. Delta A fibers are myelinated fibers, and that's why the signal is transmitted very fast without any delay. Okay, because they are covered, like insulated. Okay, and that allows them to conduct the pain and temperature signals very rapidly, fast, because the fibers are myelinated. C fibers, another type of fibers that carry the signal pain and temperature, they carry slowly because C fibers are unmyelinated. Okay, so depending on not the receptors but the fibers carrying the signal, if they are myelinated or unmyelinated. Now the hair follicles, hair receptors are attached to the hair follicles. As I showed you, this is the hair follicle and this is the hair shaft. Now this is inside the dermis, and around the hair follicle you have the efferent nerve fibers. So efferent terminal fibers enter the follicle into to encircle and form a lattice pattern around. Okay, and bending of the hair shaft would activate these receptors. Okay, I would say this is not hair shaft, around the hair follicle. This is probably typing error, hair follicle. Okay, and deflection of hair, movement of hair with your finger, soft, touch can move the hair. So that hair follicle receptors are activated by soft touch and movement of the hair in the skin where you have the hair, so hairy skin. Now here in this picture you can see those different types of receptors that I mentioned. So first here you see the Merkel's cells or endings. Okay, and Merkel cells have the vesicles filled with neuromodulators, and when these cells are activated, mechanically the rupture of the vesicles would release the chemicals into the synapse, and you see that the axon terminal and which is the afferent fiber and myelinated by the Schwann cells, covered, insulated fibers. Okay, so just know that Merkel endings are activated by the mechanical stimulus, and that causes the rupture of the vesicles and release the neuromodulators. Okay, to activate the afferent fibers. And these are slow adapting receptors. Another type of receptors that we talked about, Ruffini corpuscles or Ruffini cells. You see the nerve endings, efferent nerve endings have the receptors, and the receptors or efferent nerve endings are closely attached to the collagen fibers. So these are the collagen fibers and these are the receptors. So they are in between the collagen fibers. So when the stretch pressure is given, the collagen fibers are stretched and that activate the nerve fibers or receptors. Okay, so that is another type of slow adapting. Now let's see the corpuscles first acting. This is Pacinian. You see the nerve ending efferent and so many layers, and inside the layers you have the laminar cells. So you see these are the laminar cells, okay, and they are filled with fluid. So you have a lot of fluid inside the corpuscle, and that movement of the fluid or motion of the fluid would activate the Pacinian receptors. Okay, this is another first adapting Meissner corpuscle, first adapting type 1, and we already talked about that. The laminar cells are perpendicular to the skin. Laminar cells are in same direction, but the corpuscle is perpendicular like this, and sideways displacement of the laminar cells would activate the nerve fibers, efferent nerve fibers located in between them. Okay, here you see the free nerve endings for pain and temperature, and also another type of receptors, those are called Krauss end bulbs, and here these are actually encapsulated, and you can see the efferent nerve fiber terminals inside the capsule, and these fibers are mainly activated by the change of temperature. So these are temperature receptors. This is an interesting experiment, very simple but interesting experiment that shows why the capsule around the nerve fiber is very important. You see here, this is a Pacinian corpuscle, and you know that there are many layers around the nerve fiber, around the receptors, and this is a first adapting type 2. So when you present the stimulus, here you can see this is the Pacinian corpuscle, and this is before you present the stimulus. You do not touch the skin yet. This is the stimulus probe, and here when you press the skin, that would activate the receptors here inside the corpuscle, and you see the response immediately, right? And then the response disappears, although the stimulus probe is still there, okay, because this is first adapting, so the response is brief. Then even you kept the probe there, the response is not there anymore. Only you will see the response again when you withdraw the stimulus, you withdraw the probe. So you see the response. So this is first adapting, right? Typical first adapting type response. Then what the experimenters did, they removed the covering, the capsule. So here you see the nerve fibers, but the capsule, the layers have been removed. And now you see here, when you present the stimulus, you see the response. And as long as you keep the stimulus there, it is slow adapting. Response is still there, although gradually getting weaker, but it's still there. Okay, and when you withdraw, the response is gone. So this is a typical slow adapting type response. So now you see that after you remove the covering, the capsule, the first adapting response become slow adapting. So the capsule plays an important role to make it first adapting. Okay, without capsule, it becomes slow adapting type receptor. Transduction, the activation of the somatosensory receptors occur due to the mechanical pressure. Those are the mechanoreceptors, or thermoreceptors are activated by the temperature change, and the nociceptors can be activated by pain. And pain could be induced different ways, right? Cut, or scratch, or chemicals, whatever the stimulus is. The opening of the sodium and potassium ion channels would lead the influx of those positive ions into the efferent nerve fibers, and that will cause the depolarization of the efferent fibers. Okay, and that's how the transduction or action potential is generated. Touch receptors are pretty widely distributed in the body, and not only in the skin but other areas have the touch receptors, and more touch receptors are housed in the skin, and other areas include the mouth, within the muscles, tendons, joints. Okay. And touch receptors are present both in the epidermis and dermis, mostly in the dermis, but some Merkel cells are also located in the epidermis, okay, the deeper layer of the epidermis. This is an interesting observation. There is a test that is called the 2-point threshold discrimination test. So what the test does, you take a slide caliper which has two arms, pointed ends of two arms, and you put both pointed ends on the body surface and ask the person how many points the person feels, and gradually you increase or decrease the distance between two sharp ends. So you can increase gradually or decrease gradually, either way, or randomly you change the distance between 2 points, and touch every time, touch the skin and ask the subject how many points the subject feels. Okay, that is the minimum distance you determine when the subject can feel 2 points. Like, if first you present this stimulus, the distance is more, then decrease the distance, still the person said 2, and then you reduced further, and still the person said 2 points. That means the person can discriminate these 2 points. Now if you make it further narrow like this, very close, then the person will say, I can't tell if it is 1 or 2. The person will feel like 1 point, although there are 2 points very close to each other. So the minimum distance the subject can discriminate the 2 points, that is the 2-point threshold. So in this case, this is the threshold. Okay, and if you measure the threshold of the skin at different areas of the body, here you see this way is 2-point threshold in millimeter. That means how many millimeter is the minimum difference you can discriminate correctly, 2 points. And here you see that in some areas the person can discriminate 2 points when they are pretty close to each other, short distance. For example, in the fingertips, all fingertips have very short 2-point threshold. Okay, and then if you measure on the palm, it is more, and on the arm you see very large. The subject is not good in discriminating 2 points on the arm, or even the forearm. This is forearm, this is arm. So here again you see in the lip area, face and lip area, you have better ability to discriminate. Then in the trunk and leg you have again so weak discriminative ability. Okay, ability to discriminate. So why the fingertip and some other areas have better 2-point threshold? The reason is one I mentioned before, that is size of the receptive field. So here this is the fingertip, so the size of the receptive field is small and the receptors are more densely located. So in a small area you will find more receptors and smaller receptive fields. Now if you see here, if I use 2 points, one will activate these receptors, another point will activate this receptor, right? So 2 separate receptors are being activated and sending signal to the brain. They are kept separated, so the brain can tell that these are 2 points in the fingertip. Now let's see on the trunk. If this is your trunk surface and I put the slide caliper here, the receptors are not densely located. Okay, so even if and the size of the receptive field is big, so this is the receptive field size, for example. Okay, so these are 3 receptors. Now you see, if I use the same distance that I used for the fingertip, same receptor is being activated. So brain has no way to separate 2 points because one receptor which is being activated by both points, sending signal to the brain. Now if I increase the distance like this, then I will be able to activate 2 separate receptors, so brain will get signal separately. That's why the 2-point threshold is higher in those areas. You have larger receptive field and less densely populated neurons. This is another experiment. It is showing how the receptors on your fingertip are activated and how the maximum activation can take place. Okay, so this is a drum and on the surface of the drum you have these dots. Okay, so the drum rotates and moves downwards in like 0.2 millimeter step. So after it finishes one circle, then it will move down about 0.2 millimeter, very small, and then again after it finishes the circle it will move another 0.2 millimeter. Okay, and here you see the finger tip. Okay, and this is the receptive field of a neuron, of a receptor, and here you see different positions of the drum gradually going downwards, right, in 0.2 millimeter step. So first, for example, this dot, the top one, you use the monkey's fingertip for this experiment. So you press the fingertip against the drum, cylindrical drum, and first you place the finger in a way that this top dot touches and moves along the top part of the finger receptive field of this fingertip. Okay, so this top part only will touch the dot, and dot will move since the drum is rotating. Okay, and that will activate very little because you activated the edge of the receptive field. So you see only one firing here, only one. Okay, then you gradually move the drum down, then you move 0.2 millimeter, so it goes more inwards into the receptive field, so you can see more firing gradually. Okay, and when the single dot runs through the middle of the receptive field here, then you see 4 firings here. That means more. Single dot, remember, is passing through the middle of the receptive field, touching the middle of the receptive field. That would activate some extent. Now, when these 3 dots, this part, that means these 3 dots are moving through the middle of the fingertip or middle of the receptive field, this is number 7 here. This is the firing. You see huge increase of the firing. So that is the maximum activation, right? Because the number of dots are more, and also the dots, those 3 dots are passing through the middle of the receptive field. So that's how you can activate the receptor maximum. So that's why. And then again, when these 3 dots move towards the edge, you will see gradual decrease, and then it will go completely out, and then single dot will touch the edge, and that's why you see only one firing. So the firing or activation of the receptors would be maximum when there is stimulus is stronger. That means here 3 dots, then the 1 dot, and it is placed to the center of the receptive field. Okay. Here you see the location of the proprioceptors. Proprioceptors are located inside the muscle spindle, in the joints and tendons. Okay, so joints and tendons. So those are the locations where the proprioceptors are located. Okay. And you see here this muscle was stretched. So stretch of the muscle spindle would activate the receptor. So you see here the firing of the efferent fibers, firing rate. Okay, so pretty strong. However, if the muscle length is reduced, that means you are not stretching the muscle, you relax the muscle. You see here the muscle is relaxed like this, stretch is here, and this is relaxed condition, right? Shortening, and that would decrease the firing rate. That means the receptors are not being activated, got inactivated. Okay, so this is an experiment that can tell you that when the pressure is more, that will stretch the muscle spindle, increase the length, that would activate these efferent fibers present in the spindle. Okay, so that's how the proprioceptors are activated. Okay, and in the proprioceptors, inside the muscle spindle, you will see the afferent and efferent fibers. That makes sense, because when you stimulate, the signal is taken to the brain, right? The sensory signal. And the blue fibers here, there are two types of sensory fibers that are carrying signal from the muscle spindle to the brain, type 1A and type 2 sensory fibers. And then brain processes the signal and send motor command to the muscle to cause the contraction or movement. And the motor command arrives via the alpha motor fibers and gamma motor fibers. And you can see that alpha motor fibers are innervating the outer surface of the muscle spindle, but gamma motor fibers are going inside the muscle. Thermoreceptors are activated by the change of temperature, and as I mentioned before, that you have two types: warm fibers and cold fibers. So there are two types of fibers carry the temperature. Okay, and cold fibers are activated between the temperature 10 degrees Celsius to like 45 degrees Celsius here, and less than 10 degree or more than 40, these fibers won't respond at all, however, maximum firing or activation occurs around here, right? This is the impulses or action potentials per second. So here around 27, 26, something like that, right? Maximum activation. And the warm fibers are activated when the temperature is between 30 and 50. So if the temperature is 26 or 7, the cold fibers will be heavily activated, maximum activation, but no activation of the warm fibers. Make sense? Warm fibers are activated maximum, highest here. Make sense? So above 40, something like I can say that here it is 43 or 44 degrees Celsius. Okay, so because the fibers are two types from the receptors, the signal are being taken by that type of fibers, which is the stimulus. Okay, if it is warm, then these fibers will be activated. If it is cold, these fibers will be activated, and they will take the signal to the brain, and brain will then analyze based on the fibers brought the signal. Here you can see what I mentioned, that if you record the firing of the warmth fibers, firing rate, and change the temperature and keep watching how the fibers produce the action potential. Here you see spontaneous baseline firing at 34, and when you increase from 34 to 36, only 2 degrees Celsius, you see increase of the firing. Then another 2 degrees, you can see much more firing here, right? And then, so you can see that how sensitive these fibers are to temperature change. Slight increase would increase the firing a lot. And then 40 degree, and here you see 42 degree, you see maximum firing in this case for this fiber. This is from one fiber. And now you see these are slow adapting. That's why, you know, as long as you present the stimulus, the firing continues. Now cold fibers firing rates here. So the temperature here is less, and you can see the temperature was presented between 24 degree and 34 degree, right? So when the temperature is high, like 34 degree, spontaneous, no action potential was added. And then you reduce the temperature. As you reduce the temperature, as you move towards cold, the firing occurs more. The activation of the receptor is more. So just opposite here, as you increase the temperature, the firing is more. Here, as you decrease the temperature, the firing increases, okay, or the firing is more. So clearly these are two different types of fibers activated by two different types of temperature, cold and warm or hot. Haptic perception, that I already explained, that when you feel something, multiple information travels to the brain. If you that I mentioned, that if you touch a cubicle of ice cube, you, without looking, you can tell that it is an ice cube. However, a number of information from the finger is going to the brain, because all different receptors are being activated by the object. If it is cold or hot, right? It is cold. The surface is smooth or rough. Surface is smooth, right? Is it watery or not? Dry? That is another thing. It is watery. Is the shape is what you can feel the edges, right? It's a square, cubicle shaped. So all those, is it hard or soft? All those information together is the haptic perception, and that we use all the time to perceive the things. So here the example. Another is also you hold the object to feel the weight, the volume, the shape. Okay. Now let's stop here. I'll finish rest part in my next lecture.